I'm the Legume Breeder, part of the Legume team here at the Land Institute. Currently, we don't have 500 men, and we don't have a budget to operate a battleship. But the three of us, Abby Hahn is here, I think, and Spencer. We've been trying to do with a modest budget about what 6 people can do. We've been accomplishing about what 3 people can do, I think. Most of our work's been focusing on legume, intermediate wheatgrass, or Kernza intercrops, specifically alfalfa, like Tim mentioned. We're really excited about the opportunity to grow Kernza on a lot of acres, especially in what we're calling these dual-purpose forage and grain systems, where we're producing a human grain for humans and we're producing leaves and stems and forage for livestock. One way to supply some of Kernza's nitrogen needs is with legume for fixation, but legumes, especially alfalfa, are really high-quality forage for animals. In Kansas, alfalfa seems to be a great companion with intermediate wheatgrass. It's an excellent nitrogen fixer. And then, most importantly, it's a crop that farmers in the area are really familiar with. They grow. It's something that they're confident in. We've been directing a project for 2 years now, collaborating with other researchers from Wisconsin and Minnesota, with grant funding from SARE and the National Alfalfa and Forage Alliance to study how specific alfalfa varieties and specific alfalfa morphologies and physiologies affect the compatibility of and productivity of these Kernza alfalfa intercrops. In addition to that, we're studying all sorts of other things. We're looking at row spacing and population density. How many rows of alfalfa do we need for the most productive Kernza grain crop that we can have? We're studying things, we're looking at nitrogen fertility. How much of the nitrogen does alfalfa supply? Does it meet the requirements of intermediate wheatgrass? And then, like Tim said, we just planted a big experiment, and I could use some help trying to figure out a name for it. It's kind of a mega experiment. We planted many different varieties of alfalfa, many different varieties of multiple species of clovers, Sandpoint, some milk vetches, and et cetera. And it's probably way bigger than we should have done and could handle collecting data from. But it's going to be exciting to see if there's things that we're missing out on besides alfalfa. We've also been working with Kansas growers to try to start establishing these intermediate wheatgrass alfalfa intercrops. And there are now currently 157 acres of Kernza alfalfa intercrop farmer fields spread across 3 different counties in Kansas. Some of them have been successful, some of them have not. And there's still a lot to learn, and I think this is going to be the case with polycultures in general. As we add more species, there's more interactions between the varieties of the 2 species, interactions with the environment and the conditions of the place, and so there's a lot to learn. On that note, an especially exciting project that just began this fall through another SARE grant to the University of Minnesota, Land Institute, and the University of Wisconsin is some on-farm Kernza plus legume intercropping research. In the last month, and also on a research station in Nebraska, with the intention of creating a network of growers that can work together to help each other and to help us learn to produce Kernza grain in polycultures in ways that are economically and ecologically successful. I don't know the exact acreage, but there are other Kernza legume farmer fields and research trials in other places, 9 states in total. There's trials in Kansas, Iowa, Nebraska, Colorado, Montana, Wisconsin, Minnesota, Illinois, and New York. And in addition, there's international trials. There's trials in Canada, Europe, Australia, and elsewhere. Lastly, I want to mention that we started a big push planning multiple trials this fall working towards the development of a new perennial grain legume crop. We're sort of heading in and deciding to work on Sandpoint, it's Onobricus vicifolia. It's a really drought-tolerant perennial forage legume. People in the U.S., Montana, Wyoming, have used it to feed cattle and graze cattle for a long time. It's really fascinating. There's a seed production industry already, and I've been learning from some of the growers up there. There's some feeding trials that have been done recently showing that pigs and chickens that eat Sandpoint grain do about the same as when they're fed soybean meal. There's no developmental abnormalities for those animals. And they have monogastric systems just like us, and it suggests that we can probably eat it and consume it as well. I'm really looking forward to finding collaborators to work with, domesticating Sandpoint as a human edible grain crop. I'm especially interested if you are a food scientist or if you work at a company like impossible meats or beyond meat, looking at the protein extrusion properties of Sandpoint, the protein qualities, as well as other nutritional qualities of the plant and of the seed. Yeah, please see me if you are interested in any way in Sandpoint. And finally, we're doing plot tours here in a little bit, but I'm also going to be afterwards from 6:45 to 7:15. We have some trials on the East Bank that are Kern's alfalfa intercropping trials, and I'll be there if anyone wants to come ask questions. It's just across the north across Waterwell Road and then down the Wahub Trail, and I'll be there. So thank you guys. And also, there's a research resident poster session later, and Abby Hahn is the research resident for the Legume program, and she'll be talking about some of the Cura clover research that she's doing.
All right, so thanks, Brandon. The Crop Protection Genetics Program, which kind of protects all the crops that are being bred from diseases, has just finished its second field season, and we're still learning a lot. So personally and professionally, this has been a year dominated by moisture and disease. Since my first child was born in February, our house has been layered with spit up and milk. And while the floors, burp cloths, and diapers at home were getting doused, so were the fields. It was too wet for my alfalfa plantings to establish last winter, and it was too wet to walk in the fields for much of the summer. We had a lot of interns who went barefoot because the mud boots kept getting stuck in the fields. So that moisture brought waves of disease over the growing season, not unlike the disease hitting our home now that we have a daughter who started daycare. So as many of you know what it's like to have a new kiddo at home, I'll tell you more about our fields here at TLI. So the high rainfall this spring prompted a lot of disease in the research plots. In perennial wheat, we saw a lot of head blight infection, which is a fungal disease that produces toxin in the grain. We tested 7 of the candidate perennial wheat varieties that Schuen's developing in the disease nursery. This nursery has all the things that are needed for young developing pathogens. It has a protected habitat with lots of food, which includes our crops, and water with sprinklers that mist for every hour all night long. So as the uniform blanket of disease engulfed the perennial wheat, we determined which lines can resist that infection even under kind of ideal and perfect conditions. Of the 7 perennial wheat lines, we found only 1 that was unable to resist disease. The other 6 had much lower infection. So the next step is going to be to send that grain to North Dakota for collaborators to test for toxin analysis. So Kernza can also be infected by that same fungal pathogen that causes head blight. And for the second year, it was kept in the disease nursery. But even under pretty ideal conditions, we only see a few plants that have high infection. And we've been kind of surprised that occasionally in other fields, growers will find high toxin levels in the grain. And so we think with the abnormally high rainfall in Salina this year, we can test a theory that may be due to the contact with the soil. So during the heavy rainfalls, it can push the plants to lay flat on the soil and have contact with the ground and more fungal spores, as well as when the stalks are harvested and laid to rest on the ground for a few days to dry. That may be when the infection happens, but because we've had so much rainfall this year, we can actually test that. So in December, I moved the first Silphium and wheatgrass plants into the new crop protection wing of the greenhouse and began setting up the lab space. Since then, we've been acquiring a lot of new equipment, setting up chambers where the plants and hopefully only the pathogens, the clave, where we can sterilize pots after we're done with experiments with pathogens, and for sanitation to contain diseases when we have outbreaks in the greenhouses, which we had one for lupins this year. We're still learning how to avoid alarms and have been through a lot of electrical and plumbing hurdles in the new greenhouse. But at the moment, everything's working and we're running successful experiments. So in those new growth chambers, Angela, who's one of the interns here for the summer and dedicated to just the crop protection genetics program, has made a really interesting observation about a rust disease that affects Silphium. So this rust reduces the head number and the head weight and the overall productivity of the Silphium plants. And we're trying to develop a process so that we can infect the plants in the greenhouse and select the ones that can resist infection so that we can use breeding to improve those populations. In the past, we've been unsuccessful in trying to infect the seedlings when using our old facilities because we didn't have the ability to control temperature and humidity and light in the shared space. And we were also kind of concerned about infecting the wrong plants and inadvertently killing important breeding germplasm. So at the end of May, we set up a new experiment and looking at active infection on living plants that were taken from the field. And after a week of placing the plants in the growth chambers, the pathogen had germinated and to the infectious stage of the pathogen, the spore stage. And it coincided with the plants also dropping clusters of these black spores on the bottom of the chamber. So that kind of observation isn't something that is known in other rust diseases, and we think it's going to be very important for producing the disease infection in the next year, not something that we could easily observe in the field. So we're now working to understand what the optimal temperature and humidity are for the requirements of timing to inoculate with that disease so that we can infect the plants and within a few weeks, instead of 1 or 2 years, figure out which ones can resist infection. David and I have started to work to determine the cause of a strange, possibly viral symptom of Silphium. The symptom seems to kind of suck the life from the plants, making the veins clear and distorting the leaves and the stalk into a wirely kind of twisted swirl. It prevents certain fields, especially if waterlogged, from making flowers. And in other fields, the symptoms have spread kind of slowly from 1 or 2 plants. And it takes maybe several years for the field to become more infected. And we've started looking for disease in the native areas as well. But we see this kind of symptom in all of the native prairies. So we've tested for every virus that we can think of that might be a good candidate. We've checked more than 20 viruses so far for sunflower, geranium, lettuce, and ornamental flowers. But we haven't found anything. They've all come back negative. So based on conversations with virologists at the Phytopathological Society and our colleague at Kansas State University, Shahida Nouri, we've decided to try a sequencing approach to identify this virus because it's probably very specific just to Silphium. And in the meantime, while we're waiting for results to come back from that, Angela has set up an experiment to try to transfer the virus from one plant to another. And we're also waiting infection on that, but it may require a special insect or even a fungus to spread that virus between plants. So this year we focused on identifying pathogens that cause the most severe diseases from last year's field season and have expanded our sampling project and to David for helping me keep research projects on track while I was out on maternity leave. And I want to thank the whole TLI community for their support of my family. They visited, brought meals, and also improved the parental leave policy. So it's a good thing. So it's made me very proud to be part of this community and very happy to work with these exceptional colleagues. So we're slowly making progress to identify some of the significant diseases and breeding crops to be able to withstand infection. But the effort has to be a lot larger to involve more researchers to help identify the diseases and then to breed the crops in different environments where we haven't grown them before, and to work with farmers and agronomists who can offer more strategies to control the disease. So very grateful to you all for your support, and look forward to talking with you more this afternoon. So thanks. Next up is David Van Tassel, who's the Silphium breeder.
I'd like to interrupt this programming for a brief message from the regulator. Turn your cell phones to silent, please. Thank you.
So in the past, I've talked to you about roots and water and caterpillars and chromosomes. And today, you've had a preview. I had to decide whether to talk about plant viruses or humans. Now, maybe this seems like an easy choice. Where's the joy in discussing selfish plant parasites that contribute nothing to the ecosystem? But frankly, viruses really aren't much better. And there's the problem that I don't think of myself as a people person. I was a shy, quiet child who enjoyed plants more than people, and my own mother worried about how I would navigate this harsh world. She came up with a brilliant solution for me. She knew that plants brought me joy and that biology was my favorite subject, so I was going to become a dentist so I wouldn't have to chit-chat with them. In Oregon, who were here for 10 months. Plenty of time to form real relationships. Not long afterwards, we switched to summer internships, and we still get truly exceptional folks thanks to Tim's recruiting efforts. But inevitably, my first real conversations with an intern happen 1 week or less before they head back to college. It's sad and disappointing for a slow social person like myself. So a couple of years ago, Tim let me experiment with an extreme internship, but you need to meet them. And you need to go see their posters, as has been mentioned. They're right behind us here. And Brandon and I will be taking them and some of the younger technicians to the National Agronomy meetings in San Antonio in November. And other things that we do with them include Aubrey organizing important discussions and training sessions. So I know that scientists are supposed to be motivated by the joy of discovery and the joy of data and number crunching. But I'll tell you that the highlights of my days this year have been my interactions with the interns, residents, and the young technicians, including Sydney, who joined the perennial oilseeds program this January after finishing a master's degree at the University of Minnesota studying Silphium. Last Prairie Festival, I had entered a 5-month period without a technician, residents, or visiting Argentinians. When Sydney and the residents showed up this winter, I realized just how social the process of science is, at least for me. Science is fueled by funding and brain power and luck, but also by human relationships, by affection, loyalty, and mutual care. Of course, I get some of that from my long-term colleagues, but the truth is that often we say hi to each other in the morning and then immediately all scatter to our own plots, our own species, our own crops. So it can be a little bit isolated for each of us. So you can probably see this theme of people, some other people stuff this year. As Catherine mentioned, several of us got to be part of a big, interesting project at the University of Kansas. It's been fun and new for me to work with folks from several disciplines and campuses, and it provided the perfect framework for several research resident projects. And if you go look at their posters or walk the Marty Bender Trail, you'll get a better idea about that project. Catherine and Ebony both have Silphium projects, and that's been a game changer. Another thing, we had about 30 people here in July for a Silphium research conference, and that wouldn't have happened 5 years ago. Aubrey and I somewhat compulsively started sending out Silphium seedlings to be planted around the country in people's backyards, farms, or community gardens. I wanted to make sense of the bewildering way that Silphium does great in unexpected places, for example, downtown St. Louis, Argentina, and Vermont, and falls apart in other places. The joy people have expressed at being included in our research and the psychological boost that gave us has been a wonderful surprise. Last year, when I was standing up here, I said that it had been the best of times and the worst of times. That is still true. But the best has been the people stuff. I'm not sure that I would have stuck it out without my Silphium sisters. I'm not joking. The great news is that affection, like viruses, can spread. And we are much more than 3 now of people who care about Silphium. And I was bound and determined to get back to viruses, in case you didn't notice that. So this touchy-feely stuff is good for the resilience of my program, but working with young researchers and artists and international collaborators and citizen scientists has made me more productive and has improved the quality and depth of our research. So I beg you to help us find funding to sustain and expand the research resident and civic science programs. If you know of people or foundations with special interests in STEM and community engagement and career development, please let me or Amy know. So I focused this talk on my direct experiences with how slow human relationship building, affection, and mutual care sustain, accelerate, and enrich my research. Not all scientists would agree, and certainly the robotic plant breeders of 2029 won't need this emotional fuel. I truly hope that the educational and outreach work I've described doesn't just help me. It's easy to argue that long-term research requires us to train and engage future researchers and the public. But please ask our residents for their honest feedback. Has this program helped them develop professionally? Does it make them more interested in building a career related to the Land Institute's work? And at the macro level, we could discuss how our scientific work is inevitably influenced by social arrangements or needs new social relationships, studies, and the source of many important
that I want to begin with is how we're beginning to create new projects interwoven with the crucial efforts in natural systems agriculture being advanced by all of my colleagues here in research and commercialization. So this summer, the Perennial Oilseeds Program and the Ecosphere Studies Program collaborated to send out Silphium seedlings impulsively but very intentionally to 40 excited people who have been growing and tending these plants in 18 states across the country. Participants in our civic science community have been sharing scientific data and measurements with us, as well as practical feedback on our pilot project design. We are learning together how to organize a community to gather information about how Silphium responds to multiple growing environments, including the pollinators and pests and pathogens in those places. And we think such information will be helpful for Silphium breeding in terms of broad adaptation. But our civic science collaborators, and we do think of this community and have designed it to facilitate collaboration, not an extractive relationship where we're sort of farming out work, but where people are joining with us in a collaborative approach to co-produce this knowledge. And these collaborators are sharing their experiences with caring for Silphium. They have to water and weed and observe the plants. They explain the Silphium project to their friends and family and neighbors. They send in photographs, they exchange emails, questions, and ideas. And in monthly surveys, we ask questions about their choices and their reflections, so we can understand if and how this repetitive, modest, hands-on, ongoing, maybe unending care work shapes their learning and understanding. Most of these pilot project participants were already familiar with the Land Institute and were strongly motivated to join the project. We had a lot more interest than we could even contain in this pilot community, so you can see why we're excited about designing a larger scale version of this. And they were strongly motivated to join because of their interest in perennial agriculture and in native pollinator-friendly plants. A majority said they want to participate and contribute to authentic research. They're concerned about the future availability of food and soil, and they want to contribute to transformative, positive change in agriculture. They actually want to do this work with us, not just learn about it or hear about it. I hope they want to donate it too, but they want to do it with us. So our questions are, will their motivations persist over time? Will they develop stronger emotional attachments to just their particular Silphium plants or to plants more broadly? Will their understanding of threshold concepts in ecological agriculture, such as perenniality and diversity, deepen? Will their direct and active involvement in research matter for their own mindsets beyond the fate of the seedlings they care for? Will their experiences and the educational materials that we provide help them perceive Silphium as a beautiful, native, ornamental garden plant? Or will they learn to socially and commercially value Silphium and see it as a next-generation food crop with ecological benefits? And if any of these changes in people happen, how will we know? And how will we know why? Answering these questions involves research methods from the arts, the humanities, the social sciences, and the natural sciences, and it really invites an integrated and transdisciplinary approach. Though it will be challenging, we are excited as we undertake this community-based work. We need to learn if and how involving a broad representation of people matters. And by broad representation, I want to specifically name groups and regions whose ways of knowing are often undervalued and who are often disconnected from scientific research. We want to know how involving this broad and accurate representation of people early and deeply in a domestication process may increase the rate and quality of that domestication. So the idea that David mentioned around human domestication and the work that I've been really compelled by recently and I'm excited to dig into is this, that we can know from the history of domestication that people and plants change together and they together change landscapes. So people and plants together is what we are thinking about here. Brandon mentioned how this past winter he and I made a trip to the West Bank in Palestine to visit our collaborator Omar Tezdel and the Makanayat Research Collective there. We visited the cave and valley where some of the earliest evidence of human domestication of wheat was found millennia ago. And we met some of the women farmers who continue to plant wheat and save its seeds today. We saw the impacts of thousands of years of agriculture there on the land and on people, the soils lost, and the struggles between societies. How people feed themselves, how we eat, shapes our social arrangements and cultural practices. Agriculture shapes our relationship to this larger living planet that we're embedded within and the dynamic soil communities we are utterly dependent upon. So in an ecospheric perspective, agriculture is not just out there in fields and environments and research plots that are separate from us. But agriculture is also in here, in our bodies, in this barn, in the breaths that we take, in the metabolic processes that are keeping us awake and attentive right now, in the words and stories and norms and values for that. There are small research plots with potential new crops for Palestine, including perennial crops that Land Institute researchers are working on here in the States. Such a persistent food culture may help the Makanayat team and their collaborators build new perennial agroecosystems through community-based domestication efforts. So if the ecological future of agriculture is to become perennial and diverse, what is required of us in social terms? This is what I've come to understand as a fundamental question of the Land Institute's Ecosphere Studies work dating back to 2015, when Wes Jackson convened the first conference here in this barn with people talking here on this stage about how the invention of new perennial grain crops grown in mixtures requires new ways of thinking and making a living within the ecosphere. Since then, we've regularly designed events to bring together researchers and teachers and students and community members to do this work of co-producing and testing intergenerational approaches to learning and, I think, unlearning. There are truths that we have to remember. There are realities that we have to accept. There are some paths that we have to give up. And there are feelings of grief and pain and anger that we have to experience instead of deny. And still, there is the wondrous fact that we are alive now and have the opportunity to learn something completely new by doing this work for which we do not know the outcome. Maybe we now have the chance as people to be courageous. Since summer 2018, our Ecosphere Studies program has designed and facilitated 7 different workshops and short courses and symposia in Kansas and Nebraska and Iowa to investigate experiential learning. Most of these events were collaborations, and our partners ranged in age and background and discipline, across history and geography to psychology to farmers and ranchers and activists and artists. We think that weekend workshops like this Prairie Festival weekend can be entry points and pivot points and points of affirmation in the journeys home that we're trying to make. But we also think that we need more and deeper and ongoing learning hubs and research clusters of innovation and integration. So I'm looking forward to experimenting with that work in the coming year and building new collaborations. I want to end with the thought that it may be that only a just and caring society that accepts limits will sustain itself long enough to bring perennial polycultures into production. If so, then I am grateful to be part of a research community here and beyond, a community of people who are learning to live into and embody that future society from the ground up. Thank you for being part of that with us here. Thank you. And now I'd like to introduce Dr. Ebony Morell, the lead scientist for crop protection ecology and the recently crowned winner of the Land Institute's first Great Land Institute Bake Off.
Thank you, Aubrey. For your typical person starting out in a new position, you might argue that it is impossible to go anywhere but up. For example, when a plant breeder domesticates a new crop where there was none before, or when Aubrey creates entire education programs out of nothing. But when you're in the field of crop protection and you purposefully go about rearing entire colonies of pests and sticking caterpillars on otherwise perfectly healthy plants, at that point the definition of up gets a little hairy. In all seriousness, though, I am very pleased with what the Crop Protection Ecology Lab has accomplished in the past year. Eddie Charamond, the CPE technician, has devoted much of the past year to learning the life cycle of Eucosma giganteana, Eucosma for short, the major specialist pest of our Silphium plants. I was skeptical when Eddie told me he thought he could rear Eucosma in the laboratory within 1 year. But thanks to his diligence, he was able to do exactly that. Rear Eucosma from caterpillars to adults on a blend of Silphium plants and artificial diet. With the work Eddie has done, we are now several steps closer to understanding the life cycle of this pest, and this will help us to develop new methods to manage this pest in the field. So thank you, Eddie, for your hard work and your perseverance. I would also like to congratulate Alex Griffin, our research resident, for her tremendous capacity for troubleshooting in science. She has taken on several rather complicated projects, one of which is investigating how Silphium plants from different parts of the country and of different lineages change in their ability to take up beneficial fungi called arbuscular mycorrhizae. I don't know what prompted her to leave New York City and travel out to the middle of Kansas to spend hours measuring plant leaves, boiling roots, and staring through a microscope to count tiny fungal structures. But I am very glad she decided to do so, and I'm immensely proud of the work she has done. For more details on Alex's work, I encourage you, as many have already, to visit the Research Residence poster session this afternoon, where she can explain the project herself and in much greater detail than I have time to disclose here. I'm excited to point out our newest member of the team, our postdoctoral associate, Dr. Chase Stratton. So Chase is a Virginia native who moved north to earn his doctorate degree at the University of Vermont. I was heartened to hear this during his interview because at that point I knew he would be able to handle Kansas weather. Beyond his tolerance for temperature extremes, Chase came to us with a wealth of knowledge in chemical ecology, particularly the characterization of plant chemicals and use of essential oils to deter pests. So now Tiffany has a partner in crime. Yet another one. Since his arrival in June, he has conducted research in perennial sorghum, studying how fertilization and mycorrhizal fungi can help to boost sorghum's defenses against insect pests. Chase will be discussing some of his research further during the tours this afternoon. I would like to thank the Perennial Sorghum Lab, the Perennial Wheat Lab, and Adam for their assistance this year in managing chinch bugs at the landscape level. Thanks to group planning and cooperation, we were able to plant the sorghum and wheat fields apart from one another and plant the spaces in between them with diverse cover crop mixtures. These practices help to reduce movement of chinch bugs from the wheat fields to the sorghum fields, where they have done tremendous damage in the past. I look forward to continuing this collaboration in future years so that we can continue to keep chinch bug damage at bay. I would also like to thank Megan and Fiona of the sorghum team for their assistance in pest spotting this year. For the second year in a row, we had a late season visit from a pest called Sorghum Midge, a tiny fly which colonizes developing sorghum seeds and destroys seed heads. Until last year, this was not even considered a pest in central Kansas. Now, thanks to climate change, it appears to be here to stay. Fortunately, Megan and Fiona spotted this fly early enough this year that we were able to keep the sorghum midge from damaging many of Stan's most valuable plants. I'm extremely proud of the work the Crop Protection Ecology Lab has done, and the progress we have made in the past year has been important. As I mentioned before, Silphium has a specialist pest, the Eucosma moth. These moths lay their eggs on provides. The next month, well before the plants were ready for harvest, I climbed into a tractor and mowed 6 of those Silphium plots down. 3 of them were experimental plots for David's breeding program. I did this because I had to. Despite our best efforts to manage the Eucosma, by August their caterpillars had colonized 60% of our flowers. If I had let those flowers mature, the caterpillars would have burrowed into the root crowns in the fall and fed there all winter. They would emerge the next year, and once again our Silphium harvest would be destroyed. At least by mowing this year's flowers, I could reduce the Eucosma and give the plants a better chance next year. But I had wanted to protect the plants this year too, and I had failed. Raina Dietrichs, our research resident and our featured artist at this year's Prairie Festival, was one of the people to come with me to the Silphium fields one night and witness the sleeping bees. I admire Raina's art and her philosophical approach to art. As I have in my science, so has Raina also encountered unexpected occurrences in her art. Clay work she has designed is dried and cracked in the sun. Animals and people mar her intricate soil tapestries. But Raina doesn't view these flaws as mistakes or failures. She considers them a part of the artwork itself and embraces the changes they wreak, even as they obscure the design she labored so intensively to create. From this confluence of accident and design arises a new work of art, unintended, but with a purpose and beauty of its own. As I looked at the Silphium stalks I had just destroyed, I thought of Raina. Then I grabbed a paper bag and walked through the field, filling it with whatever leftover oil seeds I could find. Earlier this summer, David invented a recipe for tofu made from Silphium seeds. He did, and it's delicious. He and an intern, Sienna Polk, had worked on the recipe, and I decided I would try my hand at it too. Using what I had collected, I learned to thresh and clean seeds, soak and strain them, blend and boil, press and chill. I was able to produce about a pound of Silphium tofu, which I cooked and served to our staff during the perennial wheat cook-off this year. Though the journey to get there was what I neither hoped nor intended, I was at least able to use Silphium for the purpose David has spent years domesticating it, to feed people. Through both the ups and downs, I am grateful for the team that we have built, the colleagues we work with, and the people like you who support us from afar. I thank you for your time and attention, and I look forward to reporting greater progress next year. For our next speakers, I am pleased to introduce Fiona Nabukalu, Sorghum Research Associate and Midge Spotter Extraordinaire, and the Sorghum Socialist and Director of Plant Parenthood, Stan Cox.
All right, I'll start off and then Stan will join me afterwards. So, overall, it's been a great year. We've had a lot of milestones, but the biggest highlight, and that's what I'll focus on, has been the development of rhizomatous diploid sorghum lines. So we believe that this is going to make development of perennial grain sorghum very easy. I'll let you know why this is really exciting to the sorghum breeding program. So just as a reminder, in developing perennial grain sorghum, we cross-cultivated sorghum with its perennial relative, Sorghum halepense, that's also popularly known as Johnson grass. So while cultivated sorghum is diploid, it has 20 chromosomes, the perennial parent we use is a tetraploid with 40 chromosomes. I can assure you that you're making a cross between a 20 chromosome species and a 40 chromosome species is very, very tricky and cumbersome. We've had several approaches to overcome this problem, and one of the methods that we've been using is to use synthetically induced tetraploids of cultivated sorghum. In other words, cultivated sorghum, the chromosomes of cultivated sorghum have been doubled to make it a tetraploid, in that it becomes very easy for you to make a cross with tetraploid perennial sorghum. Sorry. So we have a few of such existing lines. I mean, these have been developed by other institutes. We've tried to do it here in the past, to develop our own tetraploid cultivated sorghum, but so far we've not been so successful. It's still an ongoing experiment, but we're still working towards it. So for you to develop tetraploid sorghum, we use a chemical called colchicine, which is extracted from a lily plant. Some of you may know this chemical is toxic. It's carcinogenic and very nasty. But if you use precaution, you can double the chromosomes of cultivated sorghum, and then it becomes easy for you to cross with perennial sorghum. However, getting the tetraploids is not always guaranteed, and this explains why we only have a few existing tetraploid cultivated sorghum breeding lines that are available to us breeders. The second method that we use is to use genetic male sterility. This method is very common in hybrid seed production. I won't go so much into the details of how this works, but during meiosis, they produce unreduced gametes that can easily cross with the gametes of perennial sorghum. However, we still have limited number of genetic male sterile lines that we use in our breeding work. And as you may know, the breeders, we always need a broad spectrum of genetic diversity in our breeding programs. So much as we've been successful in developing perennial sorghum using the two above-mentioned approaches, the hybrids and their offsprings have always been tetraploids with 40 chromosomes. We've been limited in what we can cross perennial sorghum with. We just cannot introduce perenniality in any cultivated sorghum lines that we have. So our colleagues in Africa and other areas and the other collaborators that we work with have always been very excited about the perennial grain sorghum. But the problem is that it's not very easy to make crosses between perennial sorghum and our cultivated sorghum. So much as they would want to introduce perenniality in their locally existing adapted cultivated sorghum, it's not always easy for them. So getting back to our excitement, last year, by chance or by, should I say, a fluke in the sorghum reproductive system, we've obtained sorghum lines that are diploid and rhizomatous. So that makes it very easy to cross perennial sorghum with other cultivated sorghum lines. So we may not necessarily have to go through other approaches in developing perennial sorghum, but we just need to make a direct cross between cultivated sorghum and perennial sorghum. So Stan may give more details about this project and the way forward. And our colleagues who happen to be here today from the University of Georgia, that's Dr. Andrew Patterson and Wen Chen, have confirmed that these diploids are coming from a cross between tetraploid cultivated sorghum and tetraploid perennial sorghum. They're also doing a lot of genotyping work to give us more genetic insights into what's happening or what's leading to these diploid rhizomatous lines. Stan, would you want to add some more?
And thank you, Fiona, for staying up here to endure some high praise when I know you'd rather go back down there and sit down. But we have bittersweet developments. Fiona is going to be leaving us at the end of the year to move to Tifton, Georgia, where finally, at long last, she and her husband Shannon and their two daughters can all live, work, and go to school in the same zip code at the same time. It's been a long time. Since Fiona arrived here as a postdoctoral fellow in 2013, some of the most exciting developments have happened in the perennial sorghum breeding work, and the one that she just described, which I'm thinking it's probably going to be a real breakthrough. Fiona was right in the middle of that one, identifying the diploid plants, and has been in the middle of all the other ones. By my count, the years that she's been here have produced 7 research papers, either published or ready for submission to journals in a wide range of research, and another legacy, not a publication, but since 2013, Fiona and Sheila Cox have taken tetraploid interspecific recombinant inbred line population from the F2 to the F13. Now, most of you will have to take my word for it that a tetraploid interspecific recombinant inbred line population in sorghum will be a very valuable genetic resource that will make possible a lot of research on the basic genetics of perenniality and other traits in the future for any researchers who want to use it. Now, Fiona, we're working on ways that Fiona will be able to continue collaborating with us while she is in Tifton, which with a university and a USDA research station has a history of sorghum research there. So we look forward to that continuing. I am also going to be departing the sorghum project gradually in the coming months, moving over to ecosphere studies. So it'll only be a move of 200 yards or so. But I will be also sort of tapering off the work on sorghum and working with Megan Gladbach, our perennial sorghum technician, on keeping the breeding cycle going, and will until someone else takes over. So if I can direct your attention to the graph over here, the red area, the x-axis is the next, in the coming months, we'll say, is the x-axis. The red area is Fiona's and my combined efforts in perennial sorghum. Will be following that, be following the red area. The green is Megan keeping the sorghum work going during this time until eventually we restaff the sorghum project with others who can work with Megan in the project. Thanks. Oh, you want to introduce Lee?
All right. On that note, we would want to introduce Dr. Lee DeHaan, the lead scientist for cancer, intermediate wheatgrass. Thank you.
It's great to be up here to talk to you today because my bottom was getting really sore sitting over there. It's a relief to come up here and stand for a while. We're almost finished. Hang in there with us through a couple more projects that we have. As was mentioned, I lead the Kernza Domestication Program. Intermediate wheatgrass was the name of this wild perennial grass. And I'd like to make sure that I say that I wasn't the one to start this program. I actually started with the Rodale Institute and then the USDA Big Flat Plants Materials Center from 1990 to 2000. Worked on developing this wild perennial grass as a grain crop. And then we started here in about 2000 to 2003, actually began a program of taking this plant that grows leaves and stems really well, and cattle like to eat it, and that's what it's grown for in the western part of the U.S. And we're encouraging it to make a whole lot more seed that tastes a lot like wheat seed. So if you get a chance to try some of the food trucks that have Kernza or the pancakes this morning, or I think there's maybe, I don't know if there's any left, but there was flour available for purchase. It's the kind of plant that we're starting to be able to grow enough to actually do things with it and eat it and use it sort of like wheat. So it's exciting times for us as we make that transition. So this wild grass is a little bit different than like we talked about with sorghum or wheat, where we had sorghum's already a great crop, makes lots of yield. In this case, we have a grass that does all the things we wanted to do ecologically, of taking up nitrogen from deep in the soil and holding on to soil and building soil carbon and outcompeting weeds, but we just don't have enough grain produced on it. So I spend all of my time thinking about how do I increase yield, which is kind of ironic because as a grad student, I was kind of skeptical about the whole yield thing. I'll tell you a little story that kind of put me onto that. We had an economist, I don't remember the person's name, came to our department in agronomy and plant genetics to talk about the economics of plant breeding, and basically showed a graph that was the exact opposite of the red line. It went up instead, and said, so you guys working in agronomy and plant genetics have made yield go up and up and up and up, and farmers have gotten less and less and less income over time because you're making yields go higher and higher and higher. there's more and more grain, and it's worth less and less and less, and they're paying more and more money to buy the inputs to grow that, and that's why farmers are not doing well. So y'all shouldn't have made all that yield because you screwed up farming. And that was point one. Point number two was that he studied the years when farmers make a lot of money and years when farmers don't, and he found that farmers make their most income on years when the yields are the lowest, because then the price goes up more than actually the yield reduction happened. And so more evidence that yield, that we say that the plant breeders are doing it all for the farmers so they can make more money, has backfired on us. And the farmers are making less money even though we have more yield. So I kind of had that perspective, and I enjoyed kind of poking the plant breeders and saying, so are you guys making progress reducing yields so the farmers can make money again? But now I spend all of my time thinking about how are we going to make more yield on Kernza and intermediate wheatgrass, because it is the one thing that's lacking. So there's so much excitement about intermediate wheatgrass, what we market as Kernza, that people want to make it into food products, farmers want to grow it, but we see that the whole supply chain just depends on having enough of it at the cost of production to the point where everyone can actually pay the bills and take it from a field to something that we can afford to buy on a shelf. So yield, ironically, is what now I think about all the time. And I think it is what stands in our way of success right now. So I've been working very hard on this since 2003 to increase yield. And this past year, we put out a trial where we had at least 5 generations, so a starting generation and then 5 generations of selection, all in one field at the same time, planted with a planter like a farmer would plant it, combine, with a combine, just like a farmer would harvest, to see if what we've been doing has worked or not. And we've worked real hard the last week. Marty Christian's my technician, and the interns threshing and weighing seeds frantically to get some of this data analyzed. And what's exciting is that, again, the opposite direction. It didn't go down, it went up in a very straight line over time. Every generation, we increased the yield by 80 kilograms per hectare very consistently for the last 5 generations. So we're on the right direction. The good news is I've done a couple more generations beyond that. We didn't have enough seed to put them in the trial. And at the early stages, it was taking us 2 to 3 years per generation. And really to do it well may have taken 5 years, because that's how long it was taking the USDA per generation. And now we have advanced techniques where we can look at the genes that are in the plants, genetic markers, so that even at the seedling stage, we can predict the best plants. And so we can cut off another whole year, but use data from 5 or even more years to pick the best plants. And so we hope we can accelerate and at least do 1 generation per year. So let's extrapolate a little bit. Something I've been trying to teach my son who's back there not to do with data in high school biology class, that you can't go beyond the edge of the graph. You can't say it keeps on going forever over here. We're gonna do that anyway. So let's say that we can keep on making this progress of 80 kilograms per hectare per year, and what will it take to equal wheat? We would need to increase the yield by another 2,100 kilograms per hectare, which is a lot. But we're making great progress. That my calculations would say that would be about 26 years to hit the yield of wheat. Seed size, also very good progress, about 30 years to equal the seed size of wheat. Now this is really disturbing news to me, not because it's so long, it's because it's so short. So we could have something that yields as much as wheat by the time of my early 70s. And I've been thinking about Wes's quote, "If your life's work can be accomplished in your lifetime, you're not thinking big enough." So I'd been under the impression I was thinking big enough that I would never, ever get there, and I had plenty of work to do. Now it looks like this assumption may be false, but I'm somewhat relieved because the wheat breeders are also making a little progress. So their yield goes up a little bit every year. And so maybe, I'll have the work to carry me through and not make it. There are other things besides yield that's important, and Tim talked about them. We've all talked about them here today. And so, even though I've had a little fun with talking about yield today, why is it that people want to eat it today, and why is it that companies want to make food out of it? General Mills had a cereal box produced that some of you may have ordered through the mail and gotten to eat with kernza grain in it. Exciting times for us, and hopefully we'll have enough to actually have one on the shelf within a year or two. Why is that? Well, it's because of all the other benefits. So it's not just yield, right? We all know that soil quality, the soil carbon, wildlife habitat, the role that a crop can play in a farmer's overall system, providing other benefits, services, the grass that can be fed to livestock, the grass that can be grazed, and provide many contributions well beyond yield. So don't think that I'm just focused on yield, but that my job really is kind of focused there right now. I want to end with my favorite quote from Wendell Berry about the future, since I did go beyond the edge of the graph there. Wendell Berry said, "You can't know where life will take you, but you can commit to a direction." And we can't predict the details of our path to perennial grains, how long it's going to take to get there, what technologies we're going to use, but we can, at least I do, remain very certain it is the correct path to take. Thank you. So in this past year, we've had exciting development, especially for me. Up and through 2010, I was the only person working on this project anywhere in the world, aside from the earlier projects. And since then, we've had just rapid expansion to researchers all over the U.S. and world, as Brandon mentioned. Many of them are actually here with us today. Very exciting work going on everywhere, and farmers wanting to grow it. And initially, basically, if a farmer wanted to grow it, they started emailing and calling me, and if they pestered me long enough, I would send them seed to get them to stop calling. And that wasn't the best way to make decisions. But we've gone through several processes of working out better plans and approaches, and finally, this year, we've hired Tessa Peters, Dr. Tessa Peters, to join us in managing the commercialization of kernza and help farmers learn about how to grow it and connect the farmers to the processors and to the end users. And that's what she's going to tell you about, so I shouldn't steal her thunder. But we're really excited to have Tessa here on our team.
And you all might be excited because I'm the last person talking. So, I want to thank Lee, and I'm really glad that he went first because as the commercialization manager for the perennial grains at the Land Institute, I fundamentally work on kernza for most of the time, but I don't expect that to be the case forever. As you heard, there have been big breakthroughs in sorghum and perennial wheat, and so we don't expect that this will be the only time we take these perennial grains and try to get them onto people's plates. And that's really exciting. And everything that I'm doing, we are learning from. This is a giant experiment. So that's why this is part of the research talk, even though we're talking mostly about commercialization. It still fits into a research program because everything we're doing has not been done before. This really is an experiment. We will learn a lot about how to do it from this pilot program of kernza that we have going on. So 9 months ago, I came on as the commercialization manager after what Rachel has called the Wild West of kernza. As you heard Lee talking about, he basically waited until someone pestered him enough that he was tired of hearing from them and then would send them some seed. And that's kind of how Kernza got into the world and how it's going on. The growers who were growing it were risk takers. It was mostly on small acres, and it was mostly funded by philanthropic investment, which it still is to some extent, but we're starting to see that changing. Now, with my coming on board at the Land Institute, we're trying to move toward more of a proof of concept phase where we want these perennials to get to market. We want to be achieving the mission of the Land Institute as we take these grains. Will we take those larger acreages and what we learn from them and apply that when we're doing polycultures and we're bringing legumes as companion crops, or any of the other crops that we start introducing into a polyculture vision? We want to make sure that this process is perennial, that it's enduring, and that we are still ensuring that this is going to be going on for a long time. We're not just letting the crops go and then having no idea what happened when all of a sudden, everyone flees the building. Commercialization process works. Inevitably, we probably will make one or two choices that we have to redirect on, but we're trying to ensure that we are asking the right questions up front so that as we guide this commercialization process, we know that it is still working on meeting the vision of the Land Institute. And there are two kind of fundamental ways that I think we go about this. The first is very practical. It's very tactical. I'll talk about it a little bit. And then the other is very strategic and some experience. It might look like coming up with planting and harvesting and fertilization techniques with our collaborators who are in the supply chain but contracting these acres. I also do a lot of connecting the growers with the researchers. So making sure that if someone has a question about polyculture and about growing alfalfa alongside Kernza, that person's in touch with Brandon, and that those growers have access to all of the knowledge of our collaborators at the University of Minnesota or University of Wisconsin or Kernza. So I calibrate them. There are going to be unpredictable responses to new environmental pressures, disease, pests, weather, weeds. These, as David was talking about, these are slow relationships. So we have to make sure that everyone who's at the table, everyone who's interested in these perennial crops understands that this is going to take time to work all of these kinks out. We don't have the benefit of the 10,000 years behind us, but we have the urgency that it needs to take a lot less time than that to switch to perennials. And that leads us to kind of developing a strategy to address these kinds of needs. And this, to me, is sort of plays on the care work that Aubrey was talking about. We have to decide what are the right questions to guide this process of commercialization. How do we get a perennial grain to market in a way that takes care of the people that are involved in this system? It's not going to happen on its own. So we do have to think about it all the time and lead this charge by living up to our values and the mission of the Land Institute as we do it. So the questions that we're asking ourselves all as a community who are involved in commercialization are things like, how can commercial products like beer, cereal, or a loaf of bread inspire consumers? How can they inspire makers of products? How can they inspire growers to change a system of agriculture that they have a history in long term and that they already understand, and that's easier in a lot of ways because of that? How can we take markets and make sure that they intentionally support rural communities? what's required of each of us along the supply chain to create an economy that shares the values of the Land Institute. Those values being people, land, and community that all relate as members, that are not in competition, but that are cooperating. This is a process that requires care. It's slow, and it's going to need course corrections from time to time. We are trying not to overdrive the headlights. We're trying to make sure that the choices we make now are the choices that are going to ensure the success of these crops. And that can be really frustrating to any number of people along the supply chain, whether that's the researchers who are, either they could be frustrated that things are going too fast or things are going too slow. It could be the people on the end who are calling constantly saying like, well, I'm a home baker and I want 5 pounds of Kernza. So you all are very lucky. Because most people I say, it doesn't really exist yet, but we're trying to get there. So you all have the opportunity today to maybe jump the gun on that. Again, this is an experiment and it requires us to leave our expectations behind because those are based on an old way of thinking, on an annual way of thinking. I listened to Winona LeDuc give a talk last year, and she said, you can't bring your old baggage to the new economy. And that really spoke to me. I think that requires a lot of steadfastness and a lot of courage. And I think as a community, the Land Institute is up for it. So I invite you all to share in that ambition and to be inspired throughout this weekend with all of the things that you see, and particularly the research plots, which I hope you get to see, depending on the weather. But also, interact with the staff and be inspired by the Prairie, because that really is the heart of what we're trying to get to. Thank you.
Well, join me one more time in thanking our group of researchers that is inspiring to work with. Thank you for your attention during the rain. We have been studying the idea of a plot tour in the last hour and a half, and we have decided to go for it. It does look like it's clearing, but there's going to be something that disappoints 70 people on the first bus, and that is that we would like it if you would if you'd rip up your tickets and go get other tickets. And the reason is we're running about 15 minutes late, and there's quite a few less people going on these tours than we actually had built out. We were dreading the prospect of there being 70 people on each tour, but the predictions for how many people were going to be here today were quite a bit larger than at least the number of people who've signed up for bus tours or plot tours, excuse me. And there's a lot of room on subsequent groups. And so we really don't want a 70-person, I don't think you want a 70-person group going through these plot tours. It'll be much more interesting to have like a 40 or 45-person tour. And so I apologize, but it really is going to be the best way forward. For what color is the first bus? White? Red. It's the bus that left at 3:43. It is white? White. Okay, so if you have a white ticket, if you wouldn't mind going back to the registration table, and there's many other colors to choose from, then you will be on a bus just a little bit later. Okay, the buses, hold on if you would please. Rachel has a couple of other important announcements. Very important announcements. But if you're going to be going on a plot tour, the first one will leave at about 2 minutes before 4. And what color is that? Blue. We'll be leaving at 2 minutes before 4, and then every 12 minutes thereafter. And please bring a raincoat, and if you have one, be prepared for a little rain, because we can't guarantee it's not going to happen, but we're going to have a good time out there. So, Rachel, a couple of comments.
Yes. Because we won't see you again necessarily until tomorrow morning, a couple things. If you're not going on the bus tours, there are the posters. There's Raina's beautiful exhibit in the gallery. There's the Marty Bender Nature Trail, which you can find out more about in the bookstore. The food trucks will be open for dinner. Most importantly, your schedule might say musical entertainment at 7:30. And I want to tell you that that is accurate but insufficient. Eliza Gilkinson has a relevant critique of the human condition, and you simply cannot be a whole person if you don't come listen to her play tonight. 7:30, right back here. Enjoy the rest of your afternoon and all we have to offer on the campus.